So today it's my really great pleasure to introduce my dear colleague Barbara Conradt. Barbara Conradt started her career at UCLA when she did her PhD in Bill Wigner's lab working on yeast vacuole biology. Afterwards, she moved actually from UCLA to the East Coast and joined their Bob Horvitz's lab at MIT, and this was a very exciting time there at that point. So Barbara, during her postdoc, did seminal work, so by cloning and by identification of the molecular function of the most upstream gene of the cell death pathway, which is the BH3-only protein AGL1, she really contributed a big deal to the understanding how the cell death pathway is working, and the whole machinery actually is working, not only in celegans, but also in other organisms. So Barbara later on continued working around the same lines in her own lab. And here I must say that a part of having an amazing record scientifically, she actually also has quite a record of moving her freezer with the worms and her lab across the Atlantic Ocean. So she started out was a junior group leader at the Max Planck Institute for Neurobiology in Germany in Martinsried. And later on, she moved back to the East Coast, to the Dartmouth Medical School. And actually 5 years ago, she moved again from the East Coast, her freezer with the worms, back to Germany, back to Bavaria, back to Munich, 500 meters away from the institution where she was before, this time to the Ludwig Maximilians University, the biocenter, where we are really happy to have her. And so during all this time, she had done amazing science, and actually this year she was awarded. She got an award for outstanding achievements in the field of cell death by the International Cell Death Society. So research in her lab spans really topics from mitochondrial dynamics through asymmetric cell division to the understanding of how the decision between life and death is made on a single cell level in a developing organism. And actually that's the topics that we will hear today from her, and the title is The Not So Simple Regulation of a Simple Cell Death Pathway. And with this, I'm going to give it over to her.
Thank you very much, Tamara, for this really nice introduction, and I would like to thank the organizers very much for inviting me to give this talk here today. So, as Tamara mentioned already, the regulation of programmed cell death is one of the things that we work on in my lab, and I don't have to tell you that programmed cell death is a really fundamental process, and we still don't know completely the regulation of this process. And celegans has been really instrumental in defining programmed cell death and its regulation. And one reason why celegans has been so important for the cell death field is the fact that during celegans development, exactly 131 cells undergo programmed cell death, and we know exactly which cells die when and where. And I just want to blow up part of the AB lineage here so that you can appreciate actually where these 131 cells are coming from. They're actually all sort of the product of an asymmetric division. A mother cell divides to give rise to a surviving cell, and I'm losing the mouse here, and a dying cell. So these divisions are asymmetric by fate. However, they are also asymmetric by size, these divisions. And that's shown here. Here labeled in red is a mother cell which undergoes cell division and forms a larger daughter, which is the daughter that is programmed to live, and the smaller daughter, which is programmed to die. And I also want to point out that about 20 to 30 minutes after the completion of cytokinesis, the smaller cell is already a cell corpse that's being engulfed and degraded. So this is a very rapid process. So because of this invariant cell death pattern, it was possible to dissect this process genetically. And screens done in the Horvitz lab and by co-workers basically identified mutations which define the genetic pathway of programmed cell death. And those 4 genes are listed here. They act in a simple linear pathway, and the loss of AGL1, CED-4, or CED-3 blocks most, if not all, programmed cell deaths that occur. So they are pro-killer genes or pro-death genes. And CED-9, in contrast, is an anti-death gene. Loss of function mutations in CED-9 actually cause ectopic cell death. And now the cloning of these 4 genes revealed that each one of them encodes a protein with homologs in higher organisms also involved in programmed cell death. And these genes actually, or these proteins, define a conserved apoptotic cell death pathway. So AGO1 and CED9 are members of the BCL2 superfamily of cell death regulators. CED4 encodes APAF1, an adapter protein, and CED3 was the very first cell death protease or caspase identified. Now, in the next few slides, I just want to tell you what we know about how the gene products of these 4 genes actually act and interact at the molecular level. And this is based on work done in a number of labs in the field. So in cells programmed to live, CED9 is active, and it localizes to the outer mitochondrial membrane, and it binds to a dimer of CED4. And this interaction basically prevents CED4 from activating the caspase CED3, and therefore the cell lives. Now, in cells programmed to die, AGO1 is active. It also binds to CED9, and this induces a conformational change in CED9, which results in CED4 release. CED4 then forms the apoptosome, composed of 8 subunits, and this apoptosome mediates the maturation and activation of the caspase, which then is the trigger for cell death or apoptosis. Now, based on its position in the cell death pathway, AGO1 really plays a very important role as the most upstream factor in this pathway, and really it is its activity that determines whether a cell will die or not. And so one of the questions we have been interested in addressing is how is AGO1 activity regulated? And work in my lab and also other labs basically resulted in data that let us propose that AGO1 activity is regulated at the transcriptional level through lineage-specific transcription factors. And this we propose based on the following data. So transcriptional reporters of the AGO locus are on in cells programmed to die, but they are mainly off in cells programmed to live. Then second, mutations in cis-acting elements of the AGO1 locus affect AGO1 expression and the highly reproducible pattern of cell death. And finally, mutations in regulators of AGO1 transcription, so transacting factors, block cell death in specific lineages. And so basically what we propose, or how it looks like, is that rather than having one transcriptional activator of AGO1 that turns the gene on in 131 cells, we probably have a transcriptional module that is specific to each specific cell death. So it's a very sort of complex and diverse regulation upstream of AGO1. And this complexity in AGO1 transcriptional regulation, which sort of we define genetically, is really reflected by the AGO1 locus on chromosome 5. So here's the AGO1 transcription units. It's a really small transcription unit, barely 1 kb. But what you see here in green are the cis-acting elements that are required for proper AGO1 transcription. And I want to point out these elements sort of go beyond the gene encoded downstream of AGO1, and they go beyond the gene upstream of AGO1. And indicated here are also transacting factors that now act through specific cis-acting elements to control AGO1 transcription, again, in a specific cell lineage, or sometimes only in one specific cell. And for the 2 stories I want to tell you today, I'm going to focus on one lineage, and that is the NSM lineage, and in particular the death of the NSM sister cell. And in this lineage, basically 2 transcription factors regulate AGO1 transcription. It is the CES-1 Snail repressor of transcription and the daughterless arcade skewed like BHLH proteins HLH2 and HLH3, which act as activators of AGO1 transcription in this specific cell lineage. And these 2 transcription factors basically act through this cis-acting element here, which we also call region B. Now on the next few, first I'm going to introduce the NSM lineage to you, and then I summarize to you some work that has been published already on what we know about AGO1 transcriptional control in this lineage. So this is the AGO1 lineage, and at 410 minutes after the first cleavage, the NSM neuroblast here divides asymmetrically to give rise to a small cell, the NSM sister cell, which is already a dead cell 21 minutes after cytokinesis. And on the right, you have the larger daughter, the NSM, which will differentiate into a serotonergic neuron. So how is AGO1 transcription controlled in this NSM lineage? And what we found very early on is that AGO1 transcription is on in the smaller daughter that's programmed to die, and it's off in the larger daughter that is programmed to live. And again, I'm going to summarize this now with these genetic models, and I'm going to explain this to you because I'm going to use this over and over again. So here I'm going to draw genetic pathways, and I'm indicating the activity of the genes in particular cells or at particular times with this color code, and orange means low activity, and blue high, and sort of black intermediate. So I just told you already that AGO1 is off transcriptionally in the larger daughter, the NSM, and that's indicated here. So it's orange, low activity, and it's not enough to induce basically the cell death pathway and to get the caspase active. That's why the larger daughter survives. However, in the smaller daughter, AGO1 is on, it's transcribed, and that is sufficient to induce caspase activation and the death of this cell. So how is this asymmetric AGO1 transcription accomplished? And it turns out that it's done through the asymmetric presence of this repressor of AGO1, CES-1 Snail. We can find and detect CES-1 protein in the larger daughter. And we think that by being present at a good concentration here in the larger daughter, CES-1 is able to prevent the activation of AGO1 through the activator HLH2, HLH3. And therefore, AGO1 transcription stays off. However, in the smaller daughter, we never detect CES-1, so very low level, and that allows HLH2, HLH3 dependent activation of AGO1 transcription, leading to death. Now, that's really good, but you might ask, so how come that there is CES-1 in the larger daughter and not in the smaller daughter? So it must be somehow asymmetrically present or asymmetrically segregated. So what is going on in the mother cell? And I told you already these cells divide asymmetrically by fate and size. And indeed, the asymmetric division of the mother cell is really important, and it's actually required for the cell-less fate, the proper cell-less fate of the daughters. And what is amazing is that CES-1, which here directly regulates AGO1, actually plays an important role in the mother as well. And what it does there is it controls two fundamental processes. It controls cell cycle progression, and it controls global polarity. And we think that by the coordinate control of these two processes by CES-1, CES-1 is really important for the ability of the cell to divide in an asymmetric manner and to set up the symmetry or asymmetry in a way that results in CES-1 present in the large daughter, but not in the small daughter. And we know a little bit about how CES-1 does this. So we've published this a number of years ago, that by repressing the CDC25.2 gene, which is required for cell cycle progression, CES-1 affects cell cycle regulation in the NSM neuroblast. And we have recent data now that CES-1 acts on global polarity by acting and repressing the PAR1-like kinase PIG1. So CES-1 levels in the mother are really, really important, and that's why CES-1 its transcription is regulated tightly by two factors, CES-2, a BCIP transcription factor, and by DNJ11, a MIDA1 or SURF1-like chaperone. So the first layer of regulation of the NSM sister cell that is basically asymmetric AGO1 transcription here in the daughters, and this requires a very complex regulation of the asymmetry or polarity of the mother cell. Now, for this next layer of regulation of the NSM sister cell, that I need to sort of give you a little bit more background, not only on apoptosis, but also on engulfment. So this is sort of an apoptotic cell, like the NSM sister cell, and I just told you EGO1 gets activated by transcriptional upregulation that leads to the activation of the CED-3 Caspase and then cell killing. And one thing that CED-3 actually does once it's activated, it activates a lipid scramblase called CED-8, and this lipid scramblase is required for the generation of the eat-me signal, and that's basically phosphatidylserine, which is flipped from the inner to the outer leaflet of the plasma membrane. Now, this eat-me signal is recognized by neighboring cells, and especially by a scavenger receptor, CED-1, and the clustering of CED-1 activates it, and it leads then to the activation of the two parallel redundant engulfment pathways. And we heard yesterday from Ryan that maybe there's actually a third pathway that is RAP35 dependent. And the activation of these two pathways then leads to the engulfment and subsequently the degradation of the apoptotic cell. Now, I just want to point out 3 genes because I'm going to be using mutations in them. CED-3 Caspase, I told you already, when you block it, all programmed cell death is blocked. I'm also going to use CED-8 mutations. CED-8 mutations block PS flipping, so they basically abolish the eat-me signal and therefore also engulfment. And CED-1 mutations block almost completely the engulfment of the cells. Now, pretty much 15 years ago, so July 12, 2001, 2 papers were published back-to-back in Nature from the Horwitz lab and Hengartner and Schnabel lab. And in these 2 papers, they showed that phagocytosis not only takes care of removing the dead cells, but it also promotes programmed cell death. And basically what they had found is that in a cell that's compromised genetic background, when you also block engulfment, you can enhance the cell death phenotype of this compromised genetic background. And they also found that this killing function of the engulfment genes acts cell non-autonomously, so not in the dying cell, but in surrounding cells, that it acts downstream of CED9, BCL2, and potentially on CED-3 Caspase, and that it acts independently of the scramblase CED-8. Now, we wanted to look at this in the NSM lineage. So we wanted to start by unraveling this, and we asked, is there also killing function of the engulfment pathways in the NSM lineage? And what we did is the following. We scored NSM sister cell survival, so in wild-type animals, they all die. But if you knock out CED-3 completely, this is a strong loss-of-function mutation, 100% of them survive. So all cell death blocked, including the death of the NSM sister cell. If you have a hypomorphic CED-3 mutation, only 13% survive, so 87% still die. Now, if you knock out the scavenger receptor CED-1, which is required for engulfment, that has no effect on NSM sister cell survival. So this strain there's no cell death defect. However, if you combine this now with this weak CED-3 loss-of-function mutation, the number goes up from 13 to 55. So loss of CED-1 function promotes the death of the NSM sister cells, and indeed, the engulfment pathways have a killing function in this lineage as well. Next question, of course, what is this killing function? So because it had been proposed that this killing function acts on CED-3, we developed some tools for CED-3. And the first thing we used is a functional phosphate-based GFP reporter, and this reporter labels all CED-3, so pro-inactive CED-3 as well as mature active CED-3. And what we found is that in the NSM neuroblast, which is shown here, very round, so this is just at metaphase, just before division, there's actually a lot of CED-3 GFP in the mother. And now when it divides asymmetrically, but when you look at concentration at time 0 after cytokinesis, you can see that both daughters inherit CED-3 GFP. And I'll show you the quantification on the next slide. It's equal by concentration. However, when you look at time 21, and this is the time at which the NSM sister cell now already looks like a corpse by DIC, you can see that the concentration has increased in this dying cell, but it has decreased in the NSM. And this is shown here, so there's a specific increase in CED-1, CED-3 GFP in the dying daughter, and a decrease in the surviving one, and at time 21, the concentration in the dying daughter is about twice that of in the surviving daughter. Now, is this affected by CED-1 loss? So first of all, so CED-3 Caspase concentration increases specifically in this dying daughter. And now the next question, is this affected if you knock out CED-1? And this is shown here. And indeed, it is affected. This increase is delayed, and it never reaches sort of the same level as we had seen in the wild type. So at time 20 minutes after cytokinesis, there's only about a 1.3-fold increase of CED-3 GFP in the smaller dying daughter. So this told us that CED-1 contributes to this CED-3 increase specifically in the dying daughter. And so I'm going to build on a slightly different model. So there's CED-3 GFP in the mother, quite a bit. During division, this is equally by concentration inherited to both daughters, and then we see a specific increase of CED-3 GFP in the small daughter and a decrease in the large daughter. And this is at least partially dependent on CED-1 and also other engulfment genes. Now the next tool we developed was a tool that allowed us to look at CED-3 activity in a spatial manner. And we took advantage of a substrate of CED-3 that we had identified, and that is TAC1. And TAC1 is actually part of the pericentriolar material, and it associates with the centrosomes during mitosis. TAC1 is a substrate of CED-3 in vitro and in vivo. And by CED-3-dependent cleavage of TAC1, we think that TAC1 no longer can associate with the centrosomes. So the association of TAC1 with the centrosome is sort of a tool for us to look at CED-3 activity. And when we looked at the NSM neuroblast in metaphase, shown here, you can see the two centrosomes, and there is, if you can see this, there's one that's larger than the other. The ventral has more GFP associated with it, and when you quantify this about 1.3-fold, so the ratio of ventrally associated GFP TAC1 to dorsally associated GFP TAC1, you have a ratio of 1.3. And if you knock out CED-3, and we looked at actually 3 different CED-3 loss of function mutants, this ratio goes down to 1.0. You lose this asymmetry in GFP TAC1 association with the centrosomes. In addition, when we knock out or when we mutate the CED-3 cleavage site in TAC1, that also abolishes this gradient. So based on this, we propose that using GFP TAC1 as a tool, we can detect a gradient of CED-3 activity along the ventral dorsal axis of the embryo, of the NSM neuroblast. And now, is this gradient of CED-3 activity that's set up in the NSM neuroblast before it divides, is this also dependent on the engulfment genes? So we looked at CED-1 mutants, but also at CED-6 and CED-2 mutants, and you can see that in all of those mutant backgrounds, this gradient was also lost. And so yes, indeed, both engulfment pathways are required for the establishment of the CED-3 activity gradient in the NSM neuroblast. And so on top of this gradient in being able to make additional CED-3 protein here in the NSM sister cell, there is a low level probably of CED-3 activity already present in the mother, and this activity is generated into a gradient or forms a gradient just before the mother divides, and this gradient is set up in a way that the part of the mother which then forms the smaller daughter, which is programmed to die, receives more of this active CED-3. And again, the engulfment genes, and CED-1 as a representative here, are required to setting this gradient in the mother up. And so where is CED-1 in this whole process? So CED-1 is really important, I told you. The clustering of CED-1 activates it, and that activates the 2 engulfment pathways. And on the next slide, I'm just going to summarize first the localization or the clustering of CED-1 around the dying daughter. And here these purple dots are supposed to be CED-1, the receptor, and in pink here, the gradient is supposed to mean the activity of the engulfment pathways. So using a CED-1 GFP reporter that we got from Zheng Zhu's lab, we could show that indeed, as expected, CED-1 clusters around the dying NSM sister cell that leads to the activation of the engulfment pathways in these cells. and eventually the complete engulfment and degradation of the dying daughter. And this is dependent on CED-3 Caspase. It's also dependent on CED-8, the scramblase, and it's mediated by PS flipping by the known eat-me signal. However, apart from this, which was expected here, we also found that CED-1 clusters actually already around the mother cell. And that's shown here, so we're using the CED-1 GFP reporter, and I hope that you can appreciate this, that the NSM neuroblast at metaphase, there is more CED-1 GFP clustered around the dorsal part compared to the ventral part. And this asymmetry is abolished in CED-3 mutants, however, it is still there in CED-8 mutants. And this is just the quantification, so in wild type, about a 1.4 difference in enrichment, dorsal to ventral. This is abolished by the loss of CED-3, but it's not significantly affected by the loss of the scramblase. So there is CED-1 enrichment already about the mother cell, and this is dependent on CED-3, but not on CED-8. So this independence of CED-8 made us wonder about the eat-me signal, so PS, so we looked, and as a control, we looked at the NSM sister cell, and you nicely see PS flipping here using an Annexin 5 GFP line. However, there is no enrichment, no PS flipping detectable in the NSM neuroblast. So this is consistent with the independence of CED-8. So this clustering of CED-1 around the NSM neuroblast, around the dorsal side, is really independent of PS flipping. So there is enrichment of CED-1 already around the mother that probably activates the two engulfment pathways in these neighbors here. And this is dependent on CED-3, but it's independent of CED-8 and PS. And now on this slide, I'm just going to put this together, our working model right now. So there is some active caspase in the NSM neuroblast, which was a real sort of shock for us. This low level of CED-3 activity here somehow results in the clustering of CED-1 around the dorsal side of the mother, resulting in the activation of the two pathways in these neighbors. And well, actually, I forgot a step. So this active CED-3 here in the mother somehow signals to result in the clustering activation of the engulfment pathways. We don't know what the signal is, and we also don't know what the symmetry breaking event is here. So now once these pathways are activated in these neighbors, they somehow signal back in the NSM neuroblast, and this then helps to establish this gradient of CED-3 activity, which then during this asymmetric division results in more active CED-3 on top of the potential to make more CED-3 into the smaller daughter, which is programmed to die. And here the engulfment pathways play a very critical role in the establishment of this gradient, which allows this asymmetric segregation. So there are lots of questions that we haven't answered yet. We don't know what the signal is, how the mother signals to the neighbors. I told you it's not dependent on the known eat-me signal. It's not dependent on PS. We also don't know how the engulfment pathways are signaling back into the mother. We don't know how this gradient is established, and we also don't know what else might be segregated asymmetrically. But this is all new work, work for things for the future. So I sort of started this part of the talk with how do celegans pathways promote the apoptotic death of the NSM sister cell. And what I can tell you now is that by promoting the cellular polarization of the NSM neuroblast, which is required for the asymmetric segregation of apoptotic potential into the NSM sister cell. And in this context, the engulfment pathways are activated by the apoptotic pathway and CED-3 in particular. However, this activation occurs in a non-canonical manner because it is independent of CED-8 and PS. And what is this good for? We believe that the segregation of apoptotic potential contributes to the robustness of the essentially invariant pattern of cell death during C. elegans development. And so this was published recently, and we were happy about this. And so now this is this asymmetric, the segregation of this potential using, again, this other model. So what I told you is that CED-3 is already active in the mother, and I haven't told you that actually the entire pathway is on in the mother already. This then leads to the activation of the engulfment pathways in neighboring cells, the formation or the polarization of the mother, and apoptotic factors, and then this leads to the asymmetric division and more of CED-3 activity and protein eventually in the smaller daughter. And now, of course, one really important question is, so this was a real shock for me. I mean, I always thought this pathway gets specifically activated in the smaller daughter, which is programmed to die after cytokinesis. But now it's already on earlier. And I mean, we've known about non-apoptotic functions of the cell death factors for a while, but this is still in the context of cell death. So what keeps this pathway in check to not kill the mother? I mean, so somehow this has to be, how is this controlled? And that brings me sort of to the third layer of this regulation, and this is actually now not in the NSM lineage. We haven't been able to address the last part. I'm going to briefly mention in the NSM lineage for technical reasons, but we've done this in the AB, ALA, PP, AAP lineage. And what it is, there are basically microRNAs involved in the control of AGO1 post-transcriptionally. And I'm just going to zoom through this really quickly, but it all started out with us analyzing the miR35 family mutants. So you heard about miR35 family already today. It's highly expressed in embryogenesis, and these animals are also embryonic lethals. And what we found is that in these miR35 family mutants, there are actually these really large abnormal cell corpses, and we called them pancakes. And this is just the quantification here. So in wild type, this is sort of, we are scoring the normal cell death. We see the normal cell death of the first wave of cell death in wild type. No large corpses in wild type, these pancakes, but in the miR35 family mutants, on average about 4.6. And we also actually knocked out the bantam family. In those animals, or we analyzed those mutants, there are no pancakes detectable, but when you combine these two knockout, or if you knock out both families, you see an increase from 4.6 to 10.7. So there's some cooperativity between these two families. So what are these pancakes? And we lineaged them all, and what we found is that each one of those pancakes is related to a cell death. And actually, most of them are mothers of cells programmed to die. So we call them precocious cell death. And what's responsible for this? So we basically looked whether CED-3 can prevent these pancakes in the miR35 family mutants, and absolutely suppressed, as well as also in the AGO1 mutant. So basically, based on this, we believe that the miRs act upstream, most likely, of AGO1. And so, indeed, AGO1 has already been shown to be targeted on the UTR by microRNAs, and there's data from the Ambrose, Duchesne, and Rajewski lab. And there's actually was pulled down as an ELG1 target, and under the footprint of ELG1, there are two miR sites, one for the miR35 family and one for the miR58 family, and they're actually also really close to each other. So what we did is a reporter assay. We used a GFP histone fusion driven by the MAY2 promoter, which is sort of ubiquitously expressed, and we either left the MAY2 UTR, put in the AGO1 wild-type UTR, or we mutated the AGO1 UTR in the way that we mutated the miR35 site here. And what you can see here is with the MAY2 UTR, you see expression starting very early on, and this one, the lower embryo, is always just a wild-type embryo. So you can see nice expression pretty much in all cells. With the AGO1 UTR, there's no expression, and there's also, this is sort of a slight background, but there's pretty much no expression also later. Now, if you mutate the miR35 binding site here, you basically recover expression. It looks just like with the May2 UTR. And if we take the EGO1, the wild-type UTR, and cross this into the miR35 family mutant background, then we also recover expression. So basically, the EGO1 3' UTR mediates miR35 family-dependent suppression of reporter gene expression. And then we wanted to look at EGO1 transcripts using single-molecule FISH, and we looked at this cell here, ABALAPPAAP, which is a mother that dies 40% of the times in the miR35 family mutant, and actually in the miR35, miR58 double family mutant, this penetrance goes up to 80%. And we used this cell because we had a reporter and could use that to identify the cell in single molecule FISH. So what we found is the following. So this is the mother, and here you have the daughters right after division. So in a wild-type background, we cannot detect EGO1 message in the mother. And in the daughters, what we see is no message in the surviving daughter, and on average 2 messages in the dying daughter. Now, if we look at the miR35 family mutant, we now see message in the mother, and I'll show you the quantification on the next slide. And we see more message in the dying daughter, and basically in the double mir knockout, the number of messages in the mother as well as in the dying daughter goes up significantly. And this is just the quantification, and maybe just focus on this side here. This is the mother, so basically 0 copies in the wild type, and on average 1.2 copies in the miR35 family mutant. And I told you that the penetrance of precocious death of the cell in this genetic background is about 40%. And interestingly enough, about 40% of these animals that we scored here had two or more messages. So we think that 2 messages is basically above the threshold that can kill. And here you can see in the double knockout, this goes up to 5.5. There's very little effect on the messages by the mir mutant deletions in the surviving daughter, but quite a dramatic effect also in the dying daughter. And so this, just to summarize, we think that EGO1 expression is controlled in the mother by, in a cooperative manner, the miR35 and miR58 family that helps to keep EGO1 below the threshold where it can kill. And then after division, right, the control is potentially gone, but we know in this lineage then UNC3 mediated activation of EGO1 transcription really boosts EGO1 transcription, and that brings the number of EGO1 messages above this threshold that's required for killing. And so this is just to summarize again. So in the NSM lineage, we found that EGO1 transcriptional control is really, really important, this asymmetric transcriptional upregulation. Then we also discovered that on top of this, there is segregation of apoptotic potential, probably just to reinforce robustness, maybe to also give the dying daughter a kickstart, because remember, this all takes place within 20 minutes. So you have cytokinesis, and 20 minutes later, the cell is already a dead cell, which is quite an accomplishment. So maybe it also accelerates the whole process. And then we think that there is this post-transcriptional control also of EGO1 expression that acts mainly in mothers, and we hope soon to be able to look at this in the NSM lineage as well. And with this, I want to just acknowledge the people who did the work. So Shayantan Chakraborty did most of the work on this segregation of apoptotic potential, and he got help from Eric Lambie, Samik Bindu, and Tamara Mikilatsit-Vali. And Highway also helped, and the microRNA work was done by Ryan Gerard and Sebastian Lure with help from Nadine Mehmer, and we got support from lots of people in the community. And with this, I would thank you for your attention.
That was beautiful. So I have 2 questions. One is, to what extent do you think this kind of environmental, local, asymmetric cell division acting in all the other cell divisions that end up in these cell deaths? That's one question. And the other is whether you think that, if I recall correctly, another aspect of those two papers from 2001 was that the cells could live if the engulfing cell was not there. And so the question is whether it's sort of a maintenance throughout the process, that they need that communication.
So the first question, so we think that this is actually not, the interaction with the neighboring cells and with the environment is not specific to this lineage. So we've already looked at the Q lineage and we see very similar things. So we think that it might occur slightly differently in different lineages, but overall, I think this is a general phenomenon. Your second question, so they didn't show that when the engulfment cell, this was, yes, if you have a CED-3 mutation, a weak CED-3 mutation, and then you block engulfment, you sometimes see cells that start looking refractile, but then sort of return to death. So this is where I think this kicks in. So if you're not segregating, if you don't give this kickstart, but by also giving maybe some active CED-3 caspase and the potential to make more CED-3, that maybe you're not basically making it, and even if you have enough AGO1 made, maybe you don't have enough CED-3 caspase to really kill the cell. There is definitely a timing aspect too.
Thank you very much for the nice talk. I had one question and one probably incorrect but maybe interesting observation. The question is, so a couple of years ago, Ron Vale showed a really interesting case where in the Q neuroblast, there's an asymmetric division that seems to be driven by asymmetric myosin contraction in the cortex, and that if you reverse that, you can get the wrong daughter to die, which suggests that at least in that case, there's some partitioning, the partitioning itself has a direct influence on the cell death decision rather than maybe an a priori polarization. I don't know if you've looked in the neuroblast that you've studied as to whether there's also some kind of, whether it's a spindle positioning asymmetry or a myosin contractility asymmetry. And if not, then maybe does that suggest that there's a greater degree of diversity in the types of programmed cell death in neuroblasts? And then the observation, I think it noticed earlier that you showed that some of the transcriptional regulators of cell death machinery components are BHLH transcription factors, which recently in Oliver Hobart's reviews showed that those are very heavily used as modular selectors of neuroblast fate and identity, and maybe there's some overarching connection there since most asymmetric cell deaths are in neurons.
So to your first question, so we don't think that the myosin-mediated process is acting in the NSM neuroblast, but absolutely, I think this, the result is the same, that you're getting asymmetric segregation of factors. So through a different mechanism, I think, in this cell division in the Q lineage, but what we would love to do is flip the division plane, basically. I mean, we actually have mutants where the division is symmetric, and then both daughters live, but we would love to switch it, but we haven't been able to get this to work. And your second question was, right, the transcription factors. I forgot to mention that all those transcriptional regulators that have been identified so far, or most of them, have another role in that particular lineage, okay? So they're just being used, potentially in different combinations, to kill the daughter that's programmed to live. But they do have a function in the surviving daughter. So I think this is how this evolved, that whatever was used was there in the lineage already is used in a different way to then kill the smaller daughter.
Thank you.
So I was, you provoked a thought about a HAM1 from John Garriga's lab, which affected the HSN PHB neuroblast. And HAM1 physically associated with disheveled, so it suggested a possibility to fill in some of the question marks you had there, that it might be a Wnt-dependent process. Yeah, we actually have lots of hints. We know Wnt is involved, so Wnt affects the asymmetry and also the pass, and probably also RAS. Right now we can't place it, so we need to sort of develop more tools, but absolutely. And HAM1, so a number of years ago, we and also John Garriga's lab looked, and we didn't see any defects when we looked at NSM sister cell survival, but now we're going back and actually looking at the division.
And PIG1, same thing. PIG1 has pretty much no effect on NSM sister cell survival, but the division is perfectly symmetric.
Hi, regarding the asymmetry breaking, I wonder if you have ruled out the contribution of cytoskeleton molecular motors and maybe also CES-1 mRNA binding proteins.
Right, so that's a really, really good question. So we haven't looked at cytoskeletal elements. We should. What we right now think is there is a little bit more CED1 expressed in the dorsal neighbors. And it could maybe just be as simple as having more CED1 in these cells, being able to cluster quicker and more efficiently in activating first. But this is something we can also test experimentally. Cytoskeletal element, we should really look at this.
Beautiful talk, Barbara. Very briefly, I noticed your Caspase substrate reporter, the GFP, when you knocked out CED1, I think, it was evenly distributed, but the deaths affected by CED1 are very minimal. So did I miss something, or is there some substrate specificity that maybe isn't picked up by that one?
Yeah. So I think, that's a good point. I think the setting up of this gradient, again, by itself is not sufficient, disrupting it to prevent the death, only if you are already compromised for CED3. So that's why I think it's just contributing to the robustness.
Yeah, makes sense.
Okay, we're going to have to move on now. So I'd like to introduce our next speaker. Oh, thank you, Barbara.